Okay, this is chapter 14. We're going to be talking about blood in this chapter. So blood, as you remember, is a connective tissue that's suspended in a liquid matrix. It will do transportation of vital substances. It maintains the stability of your interstitial fluid, which is fluid in your tissues. It distributes heat, and the amount of blood varies with body size, so changes in fluid concentration, changes in electrolyte concentration, and amount of adipose tissue can affect it. Blood is about 8% of your body weight, and the normal amount of blood in an adult is about 5 liters. Blood cells form mostly in red bone marrow, and they're called formed elements. You have red blood cells, white blood cells, and platelets. If you were to spin your blood down, well, first put it in a vial and spin it down in a centrifuge, it would separate the blood into parts, and 55% of the blood would be plasma, 45% is red blood cells, and less than 1% is white blood cells and platelets. The percentage of red blood cells is also called hematocrit, or packed cell volume. The origin of blood cells comes from a word called hematopoiesis. This means formation of blood cells. Blood cells will originate in the red marrow from hemocytoblasts or hematopoietic stem cells. Those stem cells can then give rise to more stem cells or give rise to more specialized or differentiated cells in response to hematopoietic growth factors that will change them to lymphoid stem cells or myeloid stem cells. Lymphoid stem cells give rise to lymphocytes, and myeloid stem cells give rise to all other types of formed elements, the red blood cells and other type of white blood cells and platelets. Red blood cells are also called erythrocytes. They are a biconcave shaped cell. 1/3 of it is hemoglobin. There is, if it has oxygen attached, it's called oxyhemoglobin. If it does not, it's called deoxyhemoglobin. They lack a nucleus and mitochondria, and they cannot divide. They can produce ATP through glycolysis, which you will learn in ANP2. Here's what they look like. Red blood cell count is the number of red blood cells in a cubic millimeter or a microliter of blood. These values are probably good to know what the typical ranges are. Red blood cells counts are useful in diagnosis of disease. Changes in those counts reflect the changes in the blood's oxygen carrying capacity. The production, again, the formation of red blood cells is called erythropoiesis and occurs in red bone marrow. Low blood oxygen causes the kidneys and the livers to release a hormone called erythropoietin, which stimulates the red blood cell production. This is done by negative feedback mechanism. A hemocytoblast will then give way to the erythroblast, then reticulocyte, and then an erythrocyte. Within a matter of a few days, many new red blood cells appear in the blood. There's some dietary factors that can affect red blood cell production as well. Vitamin B12 and folic acid are required for DNA synthesis, and they're necessary for growth and division of all cells. Iron is required for hemoglobin synthesis. Red blood cell destruction can happen naturally. Squeezing through capillaries of active tissue will damage red blood cells. Macrophages in the spleen and the liver will phagocytize these damaged red blood cells. Hemoglobin from the red blood cells is decomposed into heme and globin. Heme is then decomposed into iron and biliverdin. The iron is then made available for reuse in the synthesis of new hemoglobin or is stored in the liver as ferritin. Some biliverdin is converted to bilirubin. Biliverdin and bilirubin are secreted in bile as bile pigments. The globin is then broken down into amino acids metabolized by macrophages and released into the plasma. Here is a typical life cycle of a red blood cell. Number 1, the small intestine absorbs nutrients. Number 2, blood transports absorbed nutrients. Then at 3, those red blood cells are produced in the red bone marrow. 4, the red blood cells circulate into the bloodstream for about 120 days. That's a good value to remember. So they live about 120 days. Then macrophages will phagocytize and break down the old red blood cells. Hepatocytes will break down the hemoglobin, and then iron is recycled via the bloodstream. Biliverdin and bilirubin are secreted in bile, and then number 8 there, the bile is secreted into the small intestine. White blood cells are also called leukocytes. They protect against disease, and they're produced in red bone marrow under the control of hormones called interleukins and colony-stimulating factors. There are 5 types of red blood cells and 2 groupings. They're grouped by their appearance of cytoplasm. They're either a granulocyte or an agranulocyte. A granulocyte says that there is granular substance in the cytoplasm, and agranulocytes do not have any noticeable granules. Let's look at them. So we're going to start with granulocytes, and the first one is a neutrophil. A neutrophil is a small light purple granules in a stain. What's unique about it is there's 2 to 5 sections of the nucleus. They're also called PMNs, polymorphonuclear leukocytes. The keys to this one is 1, there's a lobed nucleus. 2, it's the first to arrive at an infection site. Also, there are strong phagocytes, and also this is a large number of your white blood cells. It will be elevated in bacterial infections. There's what neutrophils look like. You can see they're bigger than red blood cells. Eosinophils is next, and this one has a coarse granules in the cytoplasm, and they will stain red. Usually if eosinophils are present, it means you're having an allergic reaction. It defends against parasites also. So it may be elevated in parasitic worm infestations or an allergic reaction. Basophils has large granules. The stain is deep blue. Sometimes it's hard to even see the nucleus. They release histamine to stimulate inflammation, as well as heparin to stop blood from clotting. They're less than 1% of your white blood cells, so they're a very small amount. They're similar to eosinophils in size and shape. Monocytes. They're the largest of the white blood cells. They have a spherical kidney-shaped oval or lobed nuclei. This is an agranulocyte. There's no granules in the cytoplasm. Monocytes are unique because it will leave the bloodstream, and when it does, it's now a macrophage. 3 to 9% of your leukocytes are going to be a monocyte. It will live for weeks or months, and it can phagocytize bacteria, dead cells, and debris. Lymphocytes are slightly larger than a red blood cell. They're the smallest white blood cell. The thing that's unique about them is they have a huge nucleus, and it kind of covers the whole cell. It's an agranulocyte. T cells and B cells are the major types, and they're important in immunity. T cells directly attack pathogens or tumor cells, and B cells produce antibodies. So this is another big portion of your white blood cells, and they may live for years. The functions of white blood cells, diapetes is when white blood cells can squeeze between the cells of the capillary wall and leave the blood vessel, then migrate towards an infection site. Phagocytosis, we know what that is. That's the engulfing and digesting of pathogens. Neutrophils and monocytes are your most mobile and active phagocytes. An inflammatory response is going to be by the basophils, and a positive chemotaxis is an attraction of white blood cells to an infection site by chemicals released by that damaged cell. Here's a picture of a splinter going into the dermis, well, through the epidermis to the dermis. Bacterial is introduced, and so we have some neutrophils coming in to do phagocytosis. Platelets are also called thrombocytes. They're a cytoplasmic fragment of what's called a megakaryocyte. It's a type of stem cell in your red bone marrow. It's produced by a hormone. Well, it's produced by hemocytoblasts in response to the hormone thrombopoietin. It lacks a nucleus. It's less than half the size of a red blood cell. Platelet count can vary from 150,000 to 350,000 microliters of blood. It helps in hemostasis, the stoppage of bleeding in damaged blood vessels, by sticking to broken surfaces. The other thing that's important to remember is it releases serotonin, which causes smooth muscles in the walls of broken blood vessels to contract. There's platelets highlighted. Plasma. Plasma is the fluid, the liquid portion of your blood. It's clear, it's straw-colored, it's 55% of the blood volume. It's made up of 92% water. It has organic and inorganic chemicals in it. It transports nutrients, gases, hormones, and vitamins. It helps regulate fluid and electrolyte balance and maintain your pH. There are plasma proteins in your plasma, and they're typically not used as an energy source. Gases and nutrients are also in the blood. The most important one are carbon dioxide and oxygen. Some of the nutrients in the plasma is amino acids, simple sugars, nucleotides, lipids, which is fats, phospholipids, and cholesterol. There is non-protein nitrogenous substances, or NPNs. These are molecules that contain nitrogen, but are not proteins. In plasma, NPNs include urea, which is a product of protein breakdown or catabolism, uric acid, which is a product of nucleic acid catabolism, amino acids, which is a product of protein digestion, creatine, which stores energy in phosphate bonds and regenerates ATP in muscles, creatinine, which is a product of creatine metabolism, and BUN, which stands for blood urea nitrogen. This indicates the health of a kidney. If the level is too high, it indicates the kidney is not excreting urea in normal quantity due to some type of impaired renal function. Plasma contains a variety of ions called electrolytes. Since they ionize in water and can conduct electricity, they are absorbed from the intestine and released as byproducts of cellular metabolism. Electrolytes found in blood plasma are sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, and sulfate. Sodium and chloride are the most abundant electrolytes. Now we're going to talk about hemostasis. This is the stoppage of bleeding. This action will include blood vessel spasm, platelet plug formation, and blood coagulation. These mechanisms are most effective in smaller blood vessel injuries. The vascular spasm is stimulated by cutting or breaking the small blood vessel. It will cause the smooth muscle to contract rapidly. The smooth blood slows blood loss very quickly, and the ends of the vessel may close completely. It's triggered by stimulation of the blood vessel wall, pain, receptor reflexes, and response lasts a few minutes, but effects can last for 30 minutes. By that time, platelet plug is formed, blood is coagulating, and serotonin from platelets cause vasoconstriction. Here's a picture of it. This is platelet plug formation. This is triggered by an exposure of the platelets to collagen through an injury. The platelets adhere to the rough surface and form a plug. Platelet coagulation is the most effective mechanism of hemostasis. A form of a blood clot is in a series of reactions in which each step activates the next one, which is what you call a cascade. There are two different types of methods, extrinsic and intrinsic clotting mechanisms. Many chemicals are used in coagulation, and these are called clotting factors. Vitamin K is necessary for functioning of some of the clotting factors, and coagulation depends on the balance between procoagulants and anticoagulants. Major events in the conversion of soluble fibrinogen to insoluble threads of fibrin will trap blood cells. We probably won't go into that much detail about that. We're going to talk briefly about these concepts. So with extrinsic clotting mechanism, this is triggered by the blood coming in contact with tissues outside of a blood vessel or damaged vessel. Damaged tissues will release a hormone which is not found in blood. The cascade will begin involving a sequence of activities of several clotting factors. So eventually, fibrin, which is a thread-like structure, will stick to the damaged blood vessel surface and trap in the platelets. This is called a blood clot. This is an example of positive feedback mechanism. Once clotting begins, it promotes additional clotting. Intrinsic clotting mechanism can start without tissue damage. This is activated when blood comes in contact with a foreign surface, such as collagen in connective tissue instead of endothelium of a blood vessel wall. This is also triggered by a hormone found inside the body. As the extrinsic clotting mechanism begins, the sequence of activities of several clotting factors, and it results in the same mechanism, formation of a fibrin mesh or fibers, and that causes a blood clot. This is a very detailed map showing us how both extrinsic and intrinsic will end up with a clot. You do not need to know these steps. Just know some of the basics about each. You will not be tested on clotting factors. So after a clot is formed, it will retract and pull the edges of the broken blood vessel together while squeezing serum from the clot. Serum is plasma minus fibrinogen and most clotting factors. The platelet-derived growth factor stimulates smooth muscle cells and fibroblasts to repair damaged blood vessel walls, and plasmin digests fibrin threads and dissolves the blood clot. A thrombus is an abnormal blood clot that forms in a blood vessel, and an embolus is a blood clot moving through the blood vessels. A thrombosis is a blood clot in a vessel supplying a vital organ. Infarction is death of tissue which have been blocked blood vessels due to clot formation. Embolism is a blood clot that travels and then blocks a blood vessel in an organ, such as a pulmonary embolism in the lungs. Atherosclerosis is an accumulation of fat in the arterial linings and can sometimes cause abnormal clot formation. It's a common form of thrombosis. The smooth lining of blood vessels will discourage the accumulation of platelets and clotting factors. But as clots form, fibrin absorbs or latches onto thrombin and prevents the clotting reaction from spreading. Antithrombin inactivates additional thrombin by binding to it and blocking its action on fibrinogen. Some cells, such as basophils and mast cells, will secrete heparin, which is an anticoagulant. Blood transfusion and blood groups. We're going to first identify the blood groups. And there are 31 different genes known to contribute to the surface features of red blood cells, but we're going to look at only a few of the antigens. Antigens are any molecule that evoke an immune response. If an immune system finds a foreign antigen in the body, it produces antibodies against that antigen. So an antibody is proteins that react against a specific antigen. If you have incompatible blood transfusion, the donor's red blood cells will cause an immune response in the recipient, and the antibodies in the recipient's plasma agglutinate or clump the donor's red blood cells. Agglutination is clumping of the red blood cells, which can occur when an antibody in the recipient's plasma encounters a specific antigen of the donor's red blood cells. There are 33 known antigens on the red blood cell membrane, but only a few can evoke a serious transfusion reaction. Only the antigens of the ABO and the RH groups evoke serious transfusion reactions. The ABO group is based on the presence of two major antigens on the red blood cell, antigen A and antigen B. They are both carbohydrates, and antibodies are associated with some blood types. In general, a person will produce antibodies against antigens that are not present on his or her red blood cell membranes. This is a better picture than a chart, I think. So on the top left, we have type A blood. With type A blood, you have antigen A, or A antigens on that red blood cell. So then the body will produce an anti-B antibody. Opposite with type B blood, the antigen B is on the surface, and so you have anti-A antibodies. If you have type AB blood, you have both antigens with no antibodies. And if you have type O blood, you have no antigens and both antibodies. Agglutination is when those red blood cells come in contact with antibodies against them. They will clump together. Type O is called the universal donor. It lacks both A and B antigens, so type O could be donated to a person of any blood type. Often, when given to a person with different blood type, only the red blood cells are donated because plasma can contain antibodies. A type AB is the universal recipient. It lacks both anti-A and anti-B antibodies, so a type AB person can receive donor red blood cells of any type. Red blood cells also have an antigen called the Rh group. This was named after a rhesus monkey, which was first studied. The group includes several antigens and factors. The most important is antigen D. So you'll see Rh antigen or antigen D. It's the same thing. So if you're Rh positive, that means there's a presence of antigen D or other Rh antigens on the red blood cell. If you're Rh negative, then you lack the Rh antigens on the red blood cells. Anti-Rh antibodies form only in Rh negative individuals in response to the presence of red blood cells with Rh antigens. This can be serious if the Rh blood group is evident in the fetus that develops the condition erythroblastosis fetalis, or hemolytic disease of the newborn. So this is a good chart to show us how that works. If the mother is Rh negative and the fetus is Rh positive, during childbirth, the cells from the Rh positive fetus may enter into the female's bloodstream. That being the case, her body will not recognize it and start to make antibodies against that Rh positive blood cell. Now, in the future, if she has another baby with is Rh positive, those antibodies will start to attack the fetal red blood cells.